the learning French portion of it and also the actual scientific research I did while I was in Strasbourg. So first I wanted to share a bit about my journey to learning French because it was a bit non-traditional. So after graduating high school, I decided to take a gap year to learn French. I already speak Spanish because I live in the Valley and my mom is Mexican, so that's why I decided to choose French, another Romance language. And I started out, believe it or not, by doing a lot of Duolingo. I mean 2 weeks of 9 to 5 full-time Duolingo. To learn as many words as I possibly could to build a strong foundation before I took a 2-month introductory course at the IFE in Nice in the south of France. And from there I got more practice speaking, I learned a few more words, and then the natural next step for any language learner, I got a job on a sheep farm in Quebec. And that's where most of my French learning occurred actually. I spent about 4 months there and was forced to learn French in order to do my job and to communicate with the people I was living with because I lived in a francophone household and I worked full-time as a farm hand on this farm. And then I got another job on a farm, this time in Normandy in France, and it was like a campground community garden. So I got to interact with a lot of customers. I checked them in, I cleaned their compost toilets, and I set up their tents and just developed my French even more. Until my gap year was over and it was time to come back to UT. And that's when I applied to IFE. So IFE is an exchange program. It's not associated with any university, but it allows you to work full-time in an internship in whatever field you are studying. So you don't just have to be studying a language to do this. You can study a language and do molecular biology like I did. So you have to have like an intermediate level of French, around B1, and you get placed in a company and you can work for them full-time, and just that complete immersion really helped develop my French. And finally, I'm back. I'm now in Madame Pelletier's French 320E class, learning, some words that don't have to do with sheep. So my actual IFE internship was at the Institut de la Génétique et de la Biologie Moléculaire à Saint-Hilaire in Strasbourg. My PI is Christelle Bolzio, and I studied, there's one word missing for some reason, the proteins that bind to CH2-1L during neurodevelopment. So I'll explain what that means. Autism spectrum disorders are caused by a lot of different factors. They can be environmental or genetic, and these genetic factors can be point mutations, or CNVs, copy number variations, which are duplications and deletions of large portions of the chromosome. And the second most common of these CNVs is that of this region right here. So in this region is duplicated and deleted. It causes autism, but also macro and microcephaly. When we duplicated and deleted each of the 8 genes in this region, one at a time in zebrafish, which believe it or not have a very similar neurodevelopment to humans, only one of the genes in this region caused the mirror phenotype that we see in humans. So it's our hypothesis that this is the gene causing this phenotype. So my actual goal was studying the proteins that bind directly to CH2-1L to get a better idea of its interactome, the proteins it works with, and thus its role in neurodevelopment. So I got to learn a lot of new lab techniques that I could have learned in a lab in the US, but I instead raised the difficulty level to 10 and learned them in French. So I learned co-IP, Western blotting, very basic molecular biology techniques, and I actually got some results. I found that EGR2 and SOX2, which are proteins known to be involved in neurodevelopment, are bound directly to CH2-1L, which pretty strongly supports the hypothesis that CH2-1L is at least in some way involved in neurodevelopment and might even be a part of a mechanism for autism and neurodevelopmental disorders in general. So this might actually one day help develop therapies that actually improve people's quality of life. So not only did I get to do some relevant research, I also got to learn French while doing it. So I very highly recommend